name is Taylor Christ, and I'd like to welcome you to the 8th episode of Histo Helpers. Today we will be looking at components of the circulatory system. After today's lab, we hope that you are able to identify the distinctive characteristics of cardiac muscle, tell the difference between arteries, arterioles, veins, venules, and capillaries, and categorize and describe the various layers of the blood vessel wall. As usual, I would encourage you to think about how the microscopic structure of these tissues gives rise to their function in living tissue. We're going to start with slide 220, which is a section of heart stained with H&E. The heart consists of three basic layers: the epicardium, spanned by the yellow line, the myocardium, spanned by the long green line, and the endocardium, spanned by the short blue line on the right side of the slide. If you zoom back into the left side of the slide, the epicardium can be seen, again, by the yellow spanning line. It is the outermost layer of the heart and is home to dense fibrocollagenous tissue and adipose tissue, along with the coronary blood vessels and nerves that supply the heart itself. Note that no epicardial fibrocollagenous tissue is present in this section, but you will be able to see this on the glass slides in lab. You also can't appreciate the mesothelium in this section, which is a layer of simple squamous epithelial cells lining the superficial aspect of the epicardium. The mesothelium itself lines the pericardial cavity and secretes lubricating fluid into the cavity. Now that we have zoomed back out, the myocardium can be seen spanning the long green line. The myocardium consists of a thick layer of cardiac muscle tissue with characteristic striated cardiomyocytes that have been examined in previous labs. Remember, cardiomyocytes are distinguished by their centrally located nuclei, striations, intercalated discs, and branching patterns. A transverse section can be seen here next to the purple arrow, while a longitudinal section can be seen here outlined by the blue arrow. We have now zoomed in to another section of the myocardium to observe the presence of an extensive capillary network, which functions to perfuse the metabolically active cardiac muscle tissue with oxygen and nutrients. Capillaries in transverse section can be seen here next to the yellow arrows, and a longitudinal section can be seen here next to the green arrow. The walls of these small vessels are characterized by simple squamous endothelial cells without any smooth muscle or connective tissue surrounding them. We have moved to the right side of the slide to take a look at the endocardium, seen here spanning the blue line. The endocardium is the thin inner layer of the heart that lines its blood-containing chambers. The endocardium itself is composed of three distinct layers: a luminal endothelial cell layer with some subendothelial connective tissue, a middle layer consisting of loose connective tissue and some smooth muscle, and a deep subendocardial layer consisting of loose connective tissue, nerves, veins, and the cardiac conduction system. The layers of the endocardium are not well visualized in this histological section and will be examined on the next slide. We are now looking at slide 221, which is another section of heart stained with H&E. This section of the heart focuses on the sublayers of the endocardium. The endocardium spans between the orange line. The luminal endothelial cell layer is shown by the green arrow, the middle connective tissue layer is spanned by the yellow arrow, and a deeper subendothelial layer is spanned by the blue arrow. The Purkinje cells of the ventricular conduction system can be seen in the subendothelial layer and are pointed out by the peach-colored arrows. To the right, an inner portion of the myocardium is seen spanning the red line. This is slide 43, which is a section of a semilunar heart valve stained with H&E. There are two semilunar valves in the heart where blood flows from the two ventricles. The pulmonary semilunar valve is where blood flows from the right ventricle into the pulmonary trunk, and the aortic semilunar valve is where blood flows from the left ventricle into the aorta. When the ventricles of the heart contract, the pressure forces these valves open and blood flows down its pressure gradient. The valve is indicated by the orange arrows and is lined entirely by a layer of endothelial cells, which are seen at the tip of the black arrow. The core of the valve is composed of dense fibrous connective tissue, which is indicated by the light blue arrow. We're now taking a look at slide 222, which is a section of the aorta stained with Orsi fuchsin to clarify its numerous elastic fibers. The aorta receives all of the blood that is pumped out of the muscular left ventricle and therefore has a large lumen and has a thick wall full of elastic fibers and smooth muscle cells. When the heart contracts, the aorta expands to accommodate the large volume of blood rushing into it. When the heart relaxes, the semilunar aortic valve closes and the elastic tissue in the aortic vessel wall contracts to maintain adequate hydrostatic pressure. Like the heart, the aorta itself consists of three distinct layers. The outer tunica adventitia is seen spanning the green line, the middle tunica media seen spanning the blue line, and the inner or luminal tunica intima is seen at the bottom spanning the orange line. We have now zoomed into the tunica adventitia, which is composed of loosely arranged collagen fibers and elastic fibers, along with resident fibroblasts and macrophages. The blood vessels that supply the vessel walls themselves are collectively known as vasa vasorum. Vasa vasorum is pointed out in cross-section by the red arrow. There are also nerves in the tunica adventitia that supply the blood vessel walls, known as nervi vascularis, but will be seen later in the lab as they are not well visualized here. The tunica media is the middle layer, outlined in blue, consisting of smooth muscle, elastic fibers, reticular fibers, and proteoglycans. Because this slide is stained with Orsian fuchsin, you can see the elastic fibers here arranged into plates. These plates, which are also known as fenestrated elastic lamina, have holes in them like Swiss cheese to allow for the diffusion of oxygen and nutrients throughout the tunica media. The tunica media is seen just superficial to the tunica intima, which is down here at the bottom, a little bit difficult to distinguish, but is actually quite thick in elastic arteries. It is seen here spanning the orange line. The tunica intima consists of an endothelial cell layer, which is in direct contact with the lumen, along with underlying smooth muscle, collagen, and elastic fibers. We're now taking a look at slide 223, which is a section of tissue displaying several different vessels types concurrently. This slide is also stained with Orsian fuchsin to see the elastic fibers. Firstly, you can see a large muscular artery right here. It also consists of three major layers: the tunica adventitia, tunica media, and the tunica intima. Muscular arteries have more smooth muscle and less elastic fibers in relation to the larger elastic arteries, and their tunica media is quite thick in comparison to their tunica adventitia. The tunica intima is separated from the tunica media by this thick wavy line seen at the tip of the yellow arrow, called the internal elastic lamina. There is also an external elastic lamina between the tunica media and the tunica adventitia, indicated here by the brown arrow. These structures are simply concentrations of elastic tissue and are most easily identified in muscular arteries. Just like with the elastic artery, the tunica adventitia of the muscular artery contains loose connective tissue, some more sparsely arranged elastic fibers, vasa vasorum, and nervi vascularis. The tunica media is composed of numerous layers of smooth muscle, some elastic fibers, and proteoglycans. The tunica intima can barely be seen here on the luminal side of the internal elastic lamina, consisting of simple squamous endothelial cells and their constituent basal lamina. Right here next to the muscular artery, you can see a medium-sized vein. Veins function to return deoxygenated blood and waste back to the heart and lungs. Veins are distinguished by their irregularly shaped lumens and thin vessel walls. Veins contain a relatively thick tunica adventitia, a thin tunica media, and an inner tunica intima. The tunica adventitia is indicated by the yellow line and consists of loose connective tissue and some elastic fibers. Next to the red arrow, you can see some of the tunica media, which in veins is thin and contains collagen fibers, elastic fibers, and only a few layers of smooth muscle. Lastly, the tunica intima is seen at the tip of the green arrow, consisting of simple squamous endothelial cells and basal lamina. Note that veins also contain internal and external elastic lamina, such as seen in the muscular artery to the right, but are not well visualized as they are not as prominent as in muscular arteries. The structure indicated by the red arrow is a valve. Most veins have valves to prevent backflow. Veins without valves, such as the internal vertebral plexus and dural sinuses, are subject to backflow and consequently infections traveling to unwanted places. Venous systems with defective valves are also prone to backflow, which manifest as varicose veins. We've now moved to a different part of the slide to indicate a smaller sized artery here next to the green arrow. Notice the less prominent elastic lamina and the decrease in smooth muscle layers. Contrast this with a small vein seen just below it, indicated by the blue arrow. Lastly, you can see a lymphatic vessel in this slide, indicated here by the yellow arrow. Lymphatic vessels are thin-walled and function to drain lymph back from the tissues to the circulatory system. Like veins, they have valves to prevent backflow. Lymphatic vessels are identified by an irregularly shaped lumen and a lack of red blood cells in it. The function and components of the lymphatic system will be explored in much more depth next week by our very own Elliot Koo. We are now looking at slide 239, which is a transverse section of esophagus stained with H&E. We have reviewed components of this slide several times before in HistoHelpers, but today we're going to draw your attention to arterioles, which are smaller diameter vessels that extend and branch off from arteries. They have a thin tunica adventitia, a tunica media with 1 to 2 smooth muscle layers, and a tunica intima. I have pointed out several arterioles in the submucosa with green arrows. We have now moved to a different section of the slide to compare arterioles with venules. I have pointed to a venule here in blue, and an arteriole just above it in orange. Venules are small diameter components of the microcirculation that take blood from capillary networks and deliver them to veins. Similar to veins, they are irregularly shaped and have a thin vessel wall. Venules have a thin tunica adventitia, little to no tunica media, and a tunica intima composed of a simple squamous endothelial cell layer and basal lamina. On the other hand, arterioles have a circular lumen and 1 to 2 layers of tunica media smooth muscle. We're now looking at slide 224, which is a section of vena cava stained with Orsian fuchsin. The vena cava is the largest vein in the human body and has thin compliant walls that allow it to accommodate large volumes of blood. The tunica adventitia is the thickest layer in large veins and is indicated here by the yellow line. It consists of longitudinally arranged smooth muscle, loosely arranged collagen fibers, elastic fibers, and fibroblasts. I pointed out some longitudinally arranged smooth muscle here next to the pink arrows. The longitudinal muscle contracts to help with the general mechanism of venous return to the heart. The tunica media is indicated here to the right by a white spanning line, consisting only of a few layers of smooth muscle and collagen fibers. The tunica intima is seen here by the green arrow and consists of the usual layer of endothelial cells and subendothelial connective tissue. Back in the tunica adventitia, an example of vasovasorum is indicated by the red arrow. I indicated the nervi vascularis here by the blue arrow, which again are the nerves that supply the blood vessel walls. We have transitioned to slide 237, which is a section of the anterior tongue stained with H&E. At the top of the image is the dorsal aspect of the tongue, and the bottom the ventral aspect. Notice the multidirectional nature of the intrinsic tongue muscles, with its rich vascular and lymphatic supply. If I zoom in, you can get a great look at a vein at the tip of the blue arrow, some arterioles up here at the tips of the red arrows, and a nerve to the left circled in yellow. Lastly, you can see a lymphatic vessel at the tip of the green arrow. Make sure you notice the irregular shape, thin wall, and lack of red blood cells in the lymphatic vessel. Our last slide today is number 243, which is a section of the jejunum stained with H&E. If you remember, the intestinal villi are these microscopic infoldings of the mucosa, indicated here by the pointer. The core of these villi, called the lamina propria, is composed of loose connective tissue. Down the middle of the lamina propria, you can see an open dilated space called a lacteal, indicated by the green arrow. Lacteals, another name for lymphatic capillaries, are a component of the lymphatic system which function to absorb fat from your diet and deliver it to the lymphatic network, and eventually into the bloodstream. As many of you have learned, becoming comfortable with this material requires examination of many different specimens of the same tissue. The circulatory system is an important portion of histology, as it is present almost everywhere in the body and in almost every slide you look at. Try to point out components that you recognize from here on out as good practice. Well, that's a wrap for episode 8 on the circulatory system. I am absolutely thrilled to tell you that everyone's favorite histohelper, Dr. Elliot Koo, is making his return to explore the draining world of lymph tissue this week. Until next time, I thank you for watching HistoHelpers. We're here to help.